Hello, my name is Joseph Spencer and I'm an entomologist with the Illinois Natural History Survey. Today I'm going to speak to you about western corn rootworm biology and resistance management. Here's my outline. I'm going to mostly be talking about the western corn rootworm, Diabrodica virgifera. However, I will say a few words about northern corn rootworms, and a lot of their biology is very similar. I'll first talk about these animals as historic pests and talk about their biology and ecology. Then move on to talk about management history and BT, and finally talk about resistance and management. The first thing I'd like to point out is that corn rootworms are historic pests. The northern corn rootworm, pictured here on the left, is the original rootworm. It was first described in 1820 from specimens collected in Colorado. By the 1870s and 1880s, it had become a serious pest in corn in Missouri and in Illinois. The western corn rootworm, here on the Indian head penny, is a relative baby. It was described in 1867 from specimens collected in Kansas. It first became a recognized pest in 1909 when it was found feeding on sweet corn in Colorado. Unlike the northern corn rootworm, which is a native to Illinois, the western corn rootworm didn't enter Illinois until 1964. Together, these two species constitute what's often referred to as the billion-dollar insects. That's because the damage they cause and the cost to try to prevent that damage well exceeds $1 billion every year. All right, now I'd like to say something about the life cycle of these insects. And really the first point I want to make is that the biology of western and northern corn rootworms is tied to corn. Historically, the females only laid eggs in corn, and the larvae had to develop on corn roots or the roots of a few other grasses. So before I start talking about the biology, let's figure out where to start. I think starting with the eggs is a good place. On the upper right, we see some western corn rootworm eggs resting in the soil. They were laid there the previous summer, and they've spent the winter in the soil. Around May 1st, those eggs will hatch. And emerging from those eggs will be a tiny first instar or neonate western corn rootworm larva, less than 2 millimeters long. These tiny larvae, the first of 3 larval stages, are the most vulnerable. They're very small, and it's their job to find a corn root in which to begin feeding. They do this by detecting gradients of carbon dioxide in the soil that are emanating from metabolizing corn root tips. They move through existing soil pores to find these roots to begin feeding. They have a week or less to do this, or else they're going to die. If they're successful and make it to the root, the following 2 larval stages, the second and third instar, will also feed on the roots, and they may tend to feed within the roots. The final third instar causes the most damage to corn plants. They're shown here at the bottom. Adjacent to them is a picture of a severely injured corn root. This is the kind of damage that a heavy rootworm infestation can cause. Damage like this also affects yield of the plants very significantly. In fact, for every circle of root nodes destroyed by rootworm larvae, you can expect a 15 to 17% reduction in yield potential. After larvae are done feeding, they'll pupate in the soil. So this is after about 3 weeks. They'll spend a week in the soil as a pupa, shown here at the bottom of the slide. About the time that the pupae are in the soil is when we may begin to see some evidence of the heavy larval feeding in fields. That's when high winds or moist soil conditions will allow some plants that have compromised roots to begin to tip over in the field. These lodged or gooseneck plants, shown here on the left, will tip over. This interrupts the capacity of the plants to be very efficient intercepting light. It also makes it very hard to harvest these plants later in the season. And these harvest difficulties can result in further losses when you have severe rootworm infestations. The first adult rootworms will begin to emerge from the soil right around the end of June, typically June 26th. The first beetles to emerge will be the males. They emerge 5 to 7 days before the females, and they need this time to complete sexual development before they're ready to seek a mate. When the females emerge, they are sexually mature, and many will begin to release a pheromone which signals their receptiveness to males into the air shortly after they come out of the ground. Males move upwind to find these females, and the fortunate male who finds a receptive female will mate with her for 3 to 4 hours. During that time, he passes his sperm along with some accessory proteins to the female in a structure called the spermatophore, which is shown at the top of the slide. This spermatophore is really a significant investment by the male, as it requires him to produce a mass equal to about 9% of his body weight. After mating is completed, this spermatophore acts like a plug to prevent most further mating attempts. The female will then go off and begin to feed in preparation for producing some eggs. Males may go off and mate additional 2 or 3 more times. However, because of their investment, they need to feed as well in order to replenish the nutrients necessary to make the spermatophore. Now, in the days after mating, 15 to 24% of the females in the field will engage in a long-distance movement. They will actually leave the field where they emerged and fly high into the air, and may fly as far as 24 kilometers during a single flight overnight. This extraordinary ability for these insects to move is one of the reasons rootworm problems can spread so fast in the landscape. There's another interesting way that rootworms may move, and it can take them even further than 24 kilometers. During summertime thunderstorms, rootworms can be drawn into the updrafts in accompanying a thunderstorm and carried along in the storm and rained out miles and miles, even 100 miles away. These insects have been found in great heaps along the shores of Lake Michigan, where they have been rained out of a thunderstorm over the lake and accumulated along the shoreline. Well, once a female finds the field she's going to stay in, she'll begin to feed for several weeks before producing the first of 300 to 500 eggs that she'll produce during her lifespan. She'll continue to feed and lay eggs until the quality of the food available to her declines, and that usually happens shortly after the R2, R3 stage of corn development, when the corn silks begin to dry down. At that point, rootworms may be found moving outside of cornfields looking for other sources of pollen, such as weeds. However, by the time the corn matures and we approach the dent stage, most of the rootworms will have died, and the only thing remaining of the rootworm population will be their eggs in the soil, waiting to overwinter and begin a new generation the following year. What are some key points? First of all, larvae are largely restricted to feeding on corn roots. The females historically laid eggs primarily in cornfields. Most females are only mating once shortly after they emerge, and males may mate a few times. This is a very mobile group of species, especially the western corn rootworm, and we can see yield reductions of 15 to 17% per each damaged node of roots. Now, based on the rootworm dependence on corn as an egg-laying site and a larval feeding site, scientists from Illinois and Missouri, Stephen Forbes and C.V. Riley, recognized that the life cycle could be broken by not growing corn after corn. Because of this dependence, they recognized that by including a non-host plant into the rotation, something that the larvae couldn't develop on, the corn rootworm could be stopped. And so for them, recommending crop rotation was an obvious solution to the rootworm problem. The idea would be if you never grow corn after corn, you won't have a rootworm problem. Well, this would be great if we could do this. However, we recognize there are lots of reasons why we do need to grow some continuous corn. Back in the previous times, it was because you had draft animals you needed to feed on your farm, or maybe you were raising hogs or cattle. So continuous corn was very important. So the availability of insecticides following World War II really revolutionized pest control and the management of rootworm beetles. This was a way that farmers could grow continuous corn and not suffer terrible losses due to growing rootworm populations in corn on corn. This is a photo taken in El Paso, Illinois, of some people getting ready to spray some corn with DDT in 1948. However, the first rootworm trials with insecticide didn't occur in Illinois. They did occur in 1948, but they were in Nebraska. And that was the first year that insecticides were used against northern corn rootworms and western corn rootworms. What's really amazing is that in just 11 short years, in 1962, it was reported that in 1959 there was insecticide-resistant western corn rootworms in Nebraska. In the following years, those Nebraska populations became resistant to additional insecticides such as methylparathion and carbaryl, and even as recent as 2015, a new report of rootworm resistance to a pyrethroid came out of Nebraska. It was a similar story in Illinois, where beginning in 1953 we know that insecticides were being used against rootworms in our state. By 1958, there was still very excellent control of northern corn rootworms. Remember, the western didn't come into the state until 1964. However, by 1963, we had aldrin-resistant northern corn rootworms in central Illinois. So in this case, it took us no more than about 10 years for resistance to arrive in westerns and in northern corn rootworms in Illinois. By 65, there was broad resistance to both species, and in 1978, there was corn rootworm resistance to 4 insecticides in Illinois. So the rootworm's reputation for being a highly resistant species is well earned. Now, crop rotation is another management tool that rootworms have become resistant to. If you think about it, crop rotation works like a very effective insecticide that exerts really heavy selection pressure. Female rootworms whose eggs hatch in soybean fields will not leave any offspring. So rootworm beetles have been confronted with this problem, and they've evolved 2 different mechanisms to overcome crop rotation. The northern corn rootworm used its variability in egg hatch to evolve an adaptation where some of the eggs remain dormant in the soil in diapause for more than 2 years. This allows these eggs to be dormant during the period when soybeans are planted in a field and only hatch when the corn is growing in the field again. We call this prolonged diapause. The western corn rootworm, which is a more mobile species, has a different tactic. In the case of the westerns, their strong fidelity to cornfields was selected against, and the adult females would then move between corn and soybeans, laying eggs in both fields, as well as other crops like alfalfa and wheat. And this spreading of the eggs across the landscape means that some of the eggs are always going to be present in fields that will be planted to corn the following year. This was first detected in 1987 and really came to a head in 1995 when there was devastating up to 50% yield losses across Illinois and portions of Indiana. That problem with rotation resistance in the western corn rootworm beetle presented terrific challenges to corn growers. They had to protect both continuous and rotated corn. And the spread of this resistance away from the original area in Ford County out across Illinois and into surrounding states led to dramatically increased insecticide use. This insecticide use could have been perhaps reduced if there was some monitoring, because not all fields were heavily infected with rotation-resistant rootworms. A monitoring tool using sticky traps was developed by Matt O'Neill, who's now at Iowa State, when he was a graduate student here at University of Illinois. His tool was used to monitor beetles so they could be compared to an economic threshold to determine whether the resident population was likely to cause a problem when a soybean field became a first-year corn field. However, despite our best efforts to make monitoring look fun, not very many growers were doing monitoring of beetles in their soybean fields. Instead, there was a lot of soil insecticide use, so much use that it got a lot of attention from the USDA and EPA. Coincidentally, at about the same time, the first BT corn hybrids were being developed to manage rootworm populations. The incredible use of insecticide in Illinois and in surrounding states was one of the motivating factors that helped push for the commercialization of the first BT corn hybrids to manage rootworms, and that was in 2003 when Yield Guard rootworm was first commercialized. There have been multiple traits commercialized for rootworm management. 4 in particular: Cry3 BB1, Cry3A, Cry3.1AB, and Cry3435AB1. These materials came out beginning first in 2003, Cry3435 in 2016, Cry3A in 2007, and the last trait, Cry3.1AB, came out in 2014. Shortly after each product came out, it was quickly stacked with BT traits that could protect corn from lepidopteran or caterpillar pests. These are called stacked hybrids. Among the original rootworm traits, we can classify these into 2 groups. The first 3 on the list are called Cry3 toxins. They share structural similarity amongst this group. They are all based on the Cry3 toxin structure. The final one, Cry3435, has a different structure than the other 3. So while we have 4 different toxins, we really only have 2 different types or modes of action of rootworm protection. The final advancement in developing BT hybrids for rootworms began in 2010 when different rootworm BT toxins were combined in the same plants. We call these pyramided hybrids because they express multiple rootworm toxins in the same plant. Because you have 2 modes of action in the same plant, that makes them more durable because larvae need resistance to multiple BT toxins in order to overcome the BT in the plants. However, to fully exploit the benefits of pyramided hybrids, you really would like to have 2 toxins that are novel in the environment. And in the case of the BTs for corn rootworm beetles, they had all been deployed in the field with the exception of the Agrosure Duracade trait long before they were used in pyramids. With all these BTs available, it's important to have some way to organize and understand what's present in each BT hybrid. There's a handy tool for that called the Handy BT Trait Table, which you can find at the web address at the bottom of the page. This trait table lists all the traits and describes multiple packages that combine single or multiple traits for protection of corn from rootworms. They provide the names of traits that have multiple BT traits, the pyramided hybrids, as well as single corn rootworm hybrids. There's also notes provided on whether there's known resistance to the traits, and you can find this easily by searching for it online. So what are some recent trends in rootworm abundance and BT use? This is a figure showing western corn rootworm abundance in soybean fields measured in Urbana, Illinois from 1998 up until 2020. The heights of the bars are the number of western corn rootworm adults per sweep in a soybean field. And the red line shows the percent of BT corn adoption across the entire state of Illinois. You can see that western corn rootworm abundance has fluctuated a lot during the years. And even when BT adoption began to increase in the state, we didn't see much effect on the rootworm populations. It wasn't until around 2010, when the pyramided hybrids came in and we had very high levels of adoption, that maybe we're seeing reductions in populations that we could attribute to BT. I've also noted on here some years where we had very heavy rains in the spring. This can also dramatically reduce populations of rootworm beetles, particularly a year like 2015 when we almost wiped the rootworms out. I'd suggest that BT hybrids have helped reduce rootworm abundance dramatically, but they've gotten a lot of help from the weather. If we look at a broader scale pattern, we see similar trends. These are data that Kelly Estes, who is the Illinois State Agricultural Pest Survey Coordinator, these data she's collected when she has done surveys around the state of Illinois. In Kelly's surveys, she goes to multiple counties within the ag reporting districts and does sweep samples and live counts of beetles in corn and soybeans. The patterns that she shows for rootworm abundance mirror what we see in my Urbana, Illinois samples. We tend to see higher western corn rootworm populations in the northern and east central part of the state year after year. This is also true for northern corn rootworms, shown here on the right side of the figure. Their numbers have been increasing a little bit faster than westerns in recent times, with some impressive populations rebounding in the northern part of the state. These are great to sort of guide thinking about where there is a rootworm risk. However, they're not at the level where we can make an IPM decision. We need to have more finer scale monitoring for that. If you want to conduct monitoring, there are tools available. As I mentioned, there's the threshold using sticky traps to collect rootworm beetles in soybean fields as well as in corn fields. If you find on average 1.5 beetles per trap per day in soybeans or 2 beetles per trap per day in corn, that's an indication that the following year you may have an economic population of rootworm beetles in corn. A different method for monitoring rootworm abundance is doing adult counts. That's a little bit more challenging, but you essentially count all the live insects you can see moving around on corn plants. If you average more than 0.7 rootworms per plant in continuous corn, or more than half a rootworm per plant in a first-year corn field, that's an indication that you may have a problem if those fields are planted to corn the following year. There's a region-wide monitoring program carried out by Bayer, looking at beetles in corn and in soybeans. So they are carrying out the kind of monitoring that'd be great if each farmer was doing it in their own fields. So could monitoring avoid unnecessary BT use? I'd say yes. What I'm showing here are two figures. The top is the proportion of locally planted hybrids expressing BT corn rootworm traits. So these fields were sampled around my Urbana, Illinois field site. And what it shows is a very high proportion of BT corn used in rotated cornfields from 2014 all the way through 2018, with some reductions in 2019 and 2020. Below that, I'm showing the maximum numbers of rootworm beetles per sweep during the prior year. And you can see that the abundance of rootworm beetles dropped dramatically after 2015. Now, that's due to this massive flood that occurred in 2015 that almost wiped the rootworms out. But despite having historically low rootworm numbers, we didn't see a drop in the use of Bt corn hybrids. I think this was a missed opportunity to reduce the pressure on Bt corn hybrids at a time when there wasn't any sort of rootworm threat in cornfields in the area. I think the reductions in Bt use observed in 2019 and 2020 suggests the trend may be changing, and it looks like a good change. So what about Bt resistance? Well, there is rootworm resistance to Bt traits. It was first described in 2011 when Gassman indicated that in 2009 they found field-evolved resistance to the Cry3 BB1 trait in western corn rootworms in Iowa. It took just 6 years for the rootworms to become resistant to the very first trait deployed against them. In 2014 to 2016, cross-resistance among the Cry3 toxins was first demonstrated. This wasn't unexpected given that these products are structurally similar. So that means that if an insect involves resistance to one of these toxins, it also has resistance to the other two. And then in 2016 to 2019, the first confirmed reports of field-evolved resistance to Cry3435 were found in Iowa and Minnesota. Not to be left out of the resistance game, in 2019, there was the first confirmed field-evolved resistance to both Cry3BB1 and Cry3435 in North Dakota westerns and northern corn rootworm beetles. So how do we monitor and measure resistance in a population? Well, we do this using a bioassay that was developed by the Gassman lab. It's really quite simple. It's a single plant Bt resistance bioassay. What we do is we grow corn plants from various Bt hybrids and their non-Bt isolines. These plants are grown in the greenhouse until they reach an appropriate size, and then they are inoculated with individual rootworm larvae, and I put 10 larvae in each one of the cups of Bt or non-Bt corn. We then allow those larvae to develop on the corn roots, and at a fixed time after they were inoculated, we use these devices called Berlese funnels, which shine light onto the samples of root tissue and drive any surviving larvae out of the roots and into a vial of alcohol where we can observe and count and measure them. And so we measure the proportion of surviving larvae in these tests. So what do we see around Illinois? Well, I've been monitoring rootworm resistance since 2020 or since 2012, and we find that it's present in both continuous and rotated corn around the state. We have added Bt resistance documentation from DeKalb County this year from samples collected in 2019. And we've also collected new populations of rootworms from Stevenson and Warren County that we will bioassay for next year. The situation in Illinois is similar to what's found elsewhere in the Corn Belt. We have resistance to the Cry3 traits that is common in Illinois. We also know that the cross resistance is present among the Cry3 traits and that it's in both rotated and continuous corn. There is resistance to the Cry3435 AB1 trait around the Corn Belt, and we see evidence of reduced susceptibility also in Illinois, although we have limited sampling only in the eastern part of the state so far. We do know that pyramided hybrids can still perform quite well in Illinois. The key to a pyramid performing well, though, is that it expresses a Cry3435 trait, which is the only mode of action that still has good efficacy in Illinois. So what are trends in local Western corn rootworm Bt resistance? Well, I'm going to show you some information, but first I want to talk about the method that I'm going to use. I'm going to show you data for corrected larval survival on Cry3 and Cry34-35 hybrids. Now, corrected survival is calculated from a formula that is accounting for the background survival on non-Bt corn. And this quotient will range from 0 to 1, or 100%, and when it equals 1, that means that there is equal survival on both the Bt and the non-Bt hybrids. So here are data on corrected Champaign County Western corn rootworm survival in my bioassays. In this graph, the bar heights are the proportion of larval survival that's been corrected, and the years run along the x-axis. The orange bars are corrected survival for insects exposed to a Cry3 toxin, and the blue bars are corrected survival for insects exposed to Cry34-35. Looking at the orange bars first, you can see that the corrected survival for Western corn rootworms on Cry3 toxins increased from a little bit more than 45% up to 100% survival within 3 or 4 years between 2013 and 2017. This means that there is little or no remaining efficacy to Cry3 traits in Champaign County, and we have resistance to those traits. If we look at the Cry34-35 toxin, we see a slightly different story. We've had low corrected survival for populations tested on Cry34-35 for several years, with one little blip in 2015 with low replicates. We may be seeing a steady survival on these toxins, or the beginning of an uptick in survival on the Cry34. I have a feeling that while we do have good efficacy to this Cry34-35 trait, that efficacy is beginning to erode. It would be great if we could have reduced unnecessary use of Cry34-35 when we don't have economic populations. This would help us prolong the remaining efficacy of this trait. So let me close by talking about some management options. Really, rootworm resistance management is in your hands. It's important to scout and monitor your fields during the season using sticky traps or plant counts, and ask yourself questions. Are the hybrids you're using providing the efficacy that you expect? Do you need any additional treatments? Maybe you have high populations and may need to spray the corn to protect silks at pollination. Are you applying the economic threshold to your monitoring data? Does the current rootworm abundance indicate a likely threat to continuous corn or rotated corn during the next season? Doing monitoring puts a lot of power in your hands to make informed decisions. If you have an economic population that's expected, you have multiple options. Rotate to soybeans if you have a high corn rootworm population expected. This is the best way to essentially reset a field and eliminate a high rootworm population. If you must go back to corn, use a pyramided hybrid expressing Cry34-35, the trait that still has good efficacy. However, do use your experience to select traits that work in your farm. Bt resistance isn't a blanket phenomenon. There may be some traits that work on your farm. Don't be afraid to use what you know works. Also, refer to the Bt trait table to find hybrids that express traits that have performed well. If a trait isn't providing efficacy, consider a soil-applied insecticide applied to a non-corn rootworm hybrid. The soil-applied insecticide protects the root zone from corn rootworm injury. And remember, that's the way we protected corn before we had Bt. Applying a soil insecticide to a failing or failed Bt hybrid is not going to improve the resistance problem. It's only going to make it worse. When possible, rotate among multiple technologies over time. For example, a soil-applied insecticide on non-Bt corn may be appropriate for modest corn rootworm populations typical in first-year corn. higher expected populations in corn after corn may need a Bt hybrid, and that would be a pyramided hybrid. The following year after that, rotating to soybean may be needed to reset a field with a growing corn rootworm population. I emphasize monitoring data are critical to determining the likely risk level. So let me conclude with a few thoughts. Corn rootworms are historic pests with a history of resistance and a biology tied to corn. Their capacity for movement contributes to their pest status and multiple resistance. Corn rootworm monitoring is necessary to make informed IPM-based decisions about management. Intense use of Bt corn hybrids has selected for growing resistance to Bt traits across Illinois and the Corn Belt. Resistance management aims to prolong the remaining efficacy of Bt traits by rotating management technologies to limit unnecessary use of Bt traits. And finally, I want to acknowledge the groups and the people that have helped me over the years and make doing rootworm research almost enjoyable. And lastly, I'm happy to take any questions, and I'll be participating in the roundtable town hall that'll happen at some point after the meetings. So thank you for your attention. Here's my contact information. I'd love to hear from you.